take class today. Probably know why. Okay, so we continue with the with spinal cord. That's good. Do you want more land? Oh, that's okay. Okay. So we continue with the spinal cord. I haven't put the slides on the Moodle because I'm going to jump between. I haven't made like a very organized presentation, so I'm going to jump between few, and then after the talk I'll collect them and I'll put them in the Moodle. But in any case, what I think you should do for this subject is go to the textbook, because almost everything I'm going to say is organized there, and is organized there pretty well. So it's kind of I'm going to more or less repeat a lot of the stuff that is in there. So I somehow feel that it's a better reference than the presentation, in the sense that the presentation just kind of going to dive into few details a little bit more in a different way, but not much more. Okay, so the textbook. You haven't the Candel, yeah, the Candel, but try to get hold on the newest version, not the 4th edition, the 5th edition. Although, to be honest with you, the stuff that I'm going to talk to you about, I think, haven't been changed for the past 100 years or so. So probably the previous edition is just as good. Unless you're really going to study spinal cord yourself, then the details many of the details have been sorted out long time ago. Okay, so what I want to talk to you at the beginning is a little bit about motor neurons, and motor neurons are these neurons that send the axon to the muscle. We talked about the muscles. Now we're going to talk about how these muscles are being innervated. Where does this neuromuscular junction comes from? And it comes from these neurons that are called motor neurons, which are in the gray matter of the spinal cord. So the spinal cord has this kind of butterfly shape, where this is a ventral part and this is a dorsal part, and they reside in the ventral part of the gray matter of the spinal cord. And you see that they are huge. So if this is imagine this is a spinal cord of a human, and you see this is like millimeter. I mean, this is, I think, of a cat actually, but this is 1 millimeter, so it's huge neurons, really, really big neurons. And they send the dendrites. You can see if you just follow the dendrites, they're sending the dendrites everywhere. They are sending them to the dorsal part, to the white matter, very close to the midline, and they don't cross the midline, but they're very, very big, the dendrites, the neurons as well. And these were one of the reasons that these were the neurons that were studied a lot in terms of electrophysiology. So the primary, I think until the 70s or the beginning of the 80s, the main preparation that people were working on was the spinal cord of cats. They were holding the cats anesthetized, and they would open the spinal cord, and then you can access these huge neurons. And the first intracellular recording from a mammalian neuron was done by Eccles in 1953. Okay, already intracellular recording in vivo, in a living cat anesthetized, and a lot of the details of synaptic integration and dendritic function was figured out first on these neurons. So these are very important neurons. some aspects will be, I'll talk about it in a second. Some aspects are kind of, it's funny how some interesting aspects were found in motor neurons which are in a way irrelevant. But I'll tell you about it in a second. So people could actually put an electrode inside these neurons and record the inputs, and also the organization within the spinal cord helped them to perform some of the experiment. I'll touch upon that in a second. One thing I want to emphasize is that what you see here is a planar view, but they're actually kind of the radial. They're going all directions. They're going up and down and to the side. So it's like more like a ball, huge ball. And this is part of the reason while later on they became less useful, because people have started working on slices, and you can see the neurons under the microscope and you can target what you want. But then with motor neurons, if you cut a slice, you basically cut almost all the neuron. You don't leave almost much of it. So big, and the slices are like 300 microns. Okay, so this is 1 millimeter. You get a slice of 300 microns. You leave basically only the dendrites around the soma, and that's it. So you can't do much with it. And the other thing is that a lot of the interesting part comes from the fact that you can stimulate. We'll talk about it in a second. We know that the inputs are coming through the dorsal horn and going to the motor neuron. So in vivo, it's kind of easy to stick an electrode in the nerve and stimulate the input fibers, and then do stuff. In slices, it would be very difficult in the mass kind of the slice to find out what fiber would go into that. So that they became a little bit less kind of, they were left behind in a way. But people are still working on them also in slices. A lot of work is being done in turtles for some reason. I think the main reason is that you can keep these preparations for a long time in turtles. Turtles, for some metabolic reasons, they kind of they are very resistant to hypoxia, the cells, and because they can sea turtles. Because they're slow and because they live underwater, and they can spend hours underwater without breathing. So in the same way, you can take a slice of or the spinal cord of a turtle, put it in the fridge, come tomorrow, do an experiment, put it in the fridge, come tomorrow, do an experiment. So this is a very useful preparation. It's on the other hand, in cats, it was very intensive. You would have to anesthetize a cat and then work around the clock for like a week. People would kind of take shifts and kind of, because you don't want to spend the animal, so you would come, some people will do the morning, some people will do the night, and they would just take shifts until after a few days they would stop. Okay, next. What's happened? Oh, this is very intelligent, right? Sorry. Okay, so ignore this bit for a moment. We'll go back to it later on, but just about the responses of these motor neurons. So this is kind of okay. So we'll talk in a second about these. What does that IA mean? But imagine that there is like some input to this neuron that you can stimulate easy, and get synaptic input onto the neuron, which is what you can have here. And you see that the neuron resting membrane potential is around -70, and if you're not doing anything and you activate the synapses, there is some depolarization, okay, and it just lasts for some time. And now, sorry, so I don't know if everybody understand what's the situation that we're talking about now. Okay. So, do you see the rubber? Okay. good in drawing. So you have this butterfly shape of the spinal cord, and you have a motor neuron here with all its dendrites going all the way, or everywhere, okay? And through the dorsal root ganglion, there are fibers that are coming and making a synapse on the dendrite. Okay, and what we do is we put an electrode intracellularly inside these neurons. By the way, what kind of electrode do you think these are? Any idea? This is what's called sharp microelectrodes. So these are glass pipettes that are kind of what you do is actually, okay, so I'll tell you about, most of you probably haven't heard of that, so. Okay. So you take a glass pipette and you put it in a coil that heats up, and you pull it from both sides. Everybody? How come? What? But it's part of the method course, right? So not all of you have taken it? Okay. So you'll take it anyway, but I think actually in the new course they're not going to do sharp microelectrodes. So it's interesting just for the sake of the history. Okay, so you heat them up, and the glass becomes hot here, and then you pull it, okay? And then what happens is that the glass would become tapered like that, and eventually it would break, and you'll end up with two electrodes. And if you do it depending on the heat and the tension that you apply, this could be extremely sharp. This could be sub-micron, like very, very small. And then you fill the pipette with KCl. So it's the idea is that it's 3 molar of KCl. So it's like the very high concentration of potassium, because we know that the concentration of potassium inside the cell is higher than outside. And what you do is then you can take this pipette, you put a wire inside the tube, and the wire acts with a solution. So you actually have a circuit, and you get with a pipette inside the cell. Okay? In this case, you don't see the cell; you just go blind, and there are ways how to know that you actually enter the cell. I'm not going to go into that. And once you're in the cell, you can actually measure the membrane potential, meaning the difference between the potential inside the cell and outside the cell. Okay, now you put some electrode over the nerve, and you stimulate the nerve. Okay, so these axons are actually going to activate synapses on the motor neuron, and now you measure the postsynaptic response. Okay, so what you see here is you get a sub-threshold response. You don't get spikes. In this case, that on this electrode we don't put any. So on this electrode, it means that we don't put any current. We just measure the voltage. If I would give you the method course, I would kind of try to explain that. But basically, you just kind of try to measure the voltage that is coming out of here. Actually, what happens is that if you imagine this is the electrode and you have the wire, what you put here is a huge amplifier. It's a small amplifier connected to a big amplifier. That the details doesn't matter, but the idea is that you don't want to steal any current from the cell. Okay, so like if say we're going completely out of topic, but say there is an action potential here. What happens during an action potential? Sodium channels are open and injecting current into the membrane and depolarize the membrane, right? Everybody's with me here, right? Now sodium channels are tiny, and the current that they are injecting is tiny. If the current now on the membrane that is coming through the channel will have to kind of charge your amplifier in some way, there will be no action potential. There's not enough current to drive an electronic circuit here, right? what you want is that we call it an infinite resistance amplifier. So no current is going through here, almost no current, but we can still measure the voltage change. Okay, this is like an electronic trick. It doesn't matter how you get it, but that's the idea. Okay, so that's the idea of an amplifier in general. Like when you put your iPod into a hi-fi system that you want to kind of make noise, sound in all the room, you don't want the battery of the iPod to drive the speakers, because then they wouldn't move. So you have to amplify the signal and drive it with a different source. Okay, so that's the thing that happens here. Okay, so this means a zero nanoamp means that we don't apply any current. We just follow the voltage. And this is changing here. Okay, so here we inject 5 nanoamp. So now we have, in one direction we have an amplifier that measures the voltage, but in the other direction we have an amplifier that injects current. Okay, and now we're injecting 5 nanoamp into the cell, and what that does is depolarize the membrane a little bit. So you see the membrane was minus 71 or minus 72, and now it's minus 65. And now with the same input, synaptic input, we can actually get the neuron to spike. And there are two things that you can see. First of all, I have to tell you that 5 nanoamp is a huge current. 5 nanoamp in terms of neurons. If you take a cortical pyramidal cell and will drive it with 5 nanoamp, you'll blow it up. It will die immediately. There's no way it will survive it. It's a huge current. But first of all, motor neurons and motor neurons in the cat specifically are very big, so you need to use more current. That's one part of the thing. The other part of the thing is something that people often ignore, and kind of also it was basically found out after the invention of patch clamp recording. So what I told you is that these are sharp microelectrodes, and they're going inside the cell with a glass. And one reason that you can actually do these recordings is because the glass and the lipid membrane kind of like each other, and they kind of stick. But with sharp microelectrodes, they don't stick, perfectly. And what happens often is that you have like a small hole around the membrane, which means that a lot of the current that you inject immediately goes out and leaks it. That doesn't actually change the membrane potential of the neuron. And in fact, the whole, what we call input resistance of the neuron, how much current you need to drive in the neuron in order to get it to fire, is actually, these relationships are distorted. So in a way, it's hard to know, because this is kind of a sharp microelectrode recording. It's difficult to tell whether this if we need this extra depolarization for the synaptic input to drive the cell, or under normal conditions where there is no electrode in the cell, whether this thing would actually drive it to fire. Okay, there are two ways to actually tell, and specifically here, I must tell you, I don't know the answer, but there are two ways to tell. What do you think would be the ways to tell that? How would you tell if your recording is actually distorting distorting now the properties of the cell, and this is what makes the input sub-threshold. Right. So one thing to do is, instead of actually putting this electrode, you could put like a metal electrode that ends up here. Okay, and then you don't touch the cell. Okay, and what you record is only what you would see is something like that and spikes. What we've seen some of them before, right? So now you can do these extracellular recordings and give exactly the same stimulus and see whether your neuron now responding with spikes to the stimulus, and then you can start telling whether the intracellular recording distort the input. So this kind of The problem is that it's hard to do that in the same cell. So ideally what you want is to have one electrode extracellularly and then another one that will go into the cell. Then you know that all the conditions are the same, that you're stimulating the same amount of input and everything is the same. That's kind of hard. People have kind of played with that, but it actually became kind of more apparent when people started doing patch clamp. And then when you do patch clamp, what you get is a situation like that. So the membrane is kind of stuck now inside the pipette. So the pipette that you pull, actually here the hole is bigger, okay? And rather than having the whole thing go inside the membrane, what you get is a membrane getting inside the pipette and stick from the inside. And then what you get is a perfect connection here, and nothing is leaking out. So all the current that goes through the pipette, inside and outside, is directly going, nothing is going through this connection with the membrane, if you do it properly. Okay, so when people started doing this type of recording, suddenly they found out that the input resistance of the cell is much higher, something like 10 times higher than they thought it was with sharp microelectrodes. So this is kind of something to keep in mind. So it could be that these inputs are, like here, you need this extra current to get the neuron to fire, but it might not be like that. There is reason to believe why this input should trigger the neuron, and I'll show you in a second why this situation doesn't seem completely right. We'll talk about it towards the end of the talk. But what you should see is that these neurons are spiking quite a lot of spikes. This is like 2 seconds of recording. Okay, the resolution is not good enough. You can see the pixels, right? And you cannot see the individual spike on these pixels. But around, I don't know, 100 spikes in there, maybe less, a little bit, but and this relates to the thing that we talked last time, right? Because where do these spikes go to? They go to the muscle, and we talked about like how many spikes per second we need in order to activate the muscle. We need around 40, 50 spikes per second. So this is a firing rate that these neurons can produce. They can go even to higher rate if you apply more current and you depolarize them even more. And the thing that I told you that is a little bit irrelevant, but what people found out is that when you analyze these spike trains, they're kind of random. They're not very regular. It's not like every spike is coming at exactly fixed duration. Okay, and this is strange because people have started talking about coding, like whether this irregularity means something. And it's strange in the context of motor neurons, because motor neurons are obviously not coding much, they're just sending something to the muscle, and every spike causes a twitch, and there's nothing more than that in it. But it does tell you something about the way that synaptic inputs interact with spikes. Okay, so what you see here is that the inputs are coming like as a random walk type of kind of movement. So it's kind of, they're not coming like, an EPSP, and EPSP is like excitation, inhibition, and summation ends up like something that is approximated by almost white noise, what we call white noise. So it's like, it's a fluctuation, random fluctuation in a way. Some people can think about it as a diffusion process. There are a lot of works to try to understand what is the statistics of that thing. And this later on developed into more and more sophisticated studies about how integration, so it started here, but eventually this goes all the way up to the cortex. In the sense that, if you look at the synaptic inputs that are coming into a cortical neuron, they also look similar to that. And then they're talking about coding and the relation between what the fluctuation of the synaptic input has to do with the spike output becomes a more relevant question. Okay, but the first time it was actually described here. I don't know if I want to go into that. Okay, so this is another slide that what I want to mention, it's more like in the context of a method rather than a result. So what you do here is there's a cat, and you have something that moves the ankle of the cat, so it can actually move the ankle, all right, in a controlled way, and cause either flexion or extension of this joint. Okay, and the thing to understand here is a little bit different. So what you do is okay. I'll talk about it, and then we'll come back to it at the end of the talk. Okay, so what you do here is a trick that is kind of is the following. You use the electrode. So we're still in the same situation. We have an electrode in the cell. Okay, right now we're not going to stimulate the nerve with an extracellular electrode. We're going to stimulate the nerve by just kind of moving the ankle, right? And what we do at the same time is we use the electrode in the cell to change the membrane potential of the cell. Okay, and note that this time is long. This is like 3 seconds. So the idea is that you inject current and you depolarize the cell, then you inject a little bit more current and you depolarize the cell. Okay, and just like we did here, from here to here, we just do it gradually. Okay, so we go up and then we go down and then we stop and then we go up again and go down again. Okay, and what that makes, the result is because we're doing it very slowly, the voltage in the cell is actually following more or less the voltage in the neuron. Okay, the voltage is more or less following the current, and what you see is these kind of triangles response in the voltage. And now, sorry, yeah, you're right. So it's okay, right? Sorry, it's true. It's true, but it's kind of okay. So the idea, this is kind of a voltage clamp, right? You're right. I'm sorry. This is kind of a voltage clamp. You kind of clamp the voltage to a specific voltage, and you measure what current do you need to inject that. But you have to remember that that's the thing that I wanted to avoid. Okay. Did Dan talk to you about voltage clamp in dendrite? You had lectures by Dan, right? And did he talk to you about voltage clamp in dendrite? No. Okay. Anyway, you learned something from it. I don't know if it's exactly what I wanted, but maybe it's more important actually. Okay, so voltage clamp, you know what voltage clamp is, right? You learned about Hodgkin-Huxley, and you remember this idea that you have a feedback loop, that when voltage change, you inject current. Okay, so one of the things that Idan did talk to you about cable theory, right? Okay, so what actually started cable theory was exactly this kind of experiment here, when there was these motor neurons, and they had an electrode in the motor neuron, and they would kind of stimulate this IA fiber. So this would be stimulated by an electrode, and they would just record like an EPSP. Okay. And this was done in this lab of Eccles, and there was this guy, Wilfried Roll, that was a physicist that was sitting in this lab and was trying to kind of model that. And the conclusion that he got to is that the dendrites are very important. So that if the input is coming to here, or the input is coming to here, this will change the shape of the EPSP. In one case, you'll get something like that, and in the other case, you'll get something. I'll draw it more precisely. If the input coming straight to the soma, you'll get something like that, and if it will get to the dendrite, you'll get something like that. Okay, and he kind of modeled that and developed all this cable theory and stuff. Now the result, the conclusion from the other conclusion from that is that if you try to do voltage clamp here, okay, and you imagine that as a cable, so this is a soma, and say we're just looking at one dendrite now to the side. So now I want the voltage here to be minus 30. Okay, so I inject a current with my electrode. I have a current source that goes through the electrode to the cell body. Okay, sorry, like that. I inject current to go to minus 30 millivolts, but the profile of the voltage would be that here will have minus 30 millivolts, but to the side it will decay. Okay, so if I want to make the synapse kind of have minus 30 millivolts here in the dendrite, I need here to go much higher. Okay, so the voltage clamp. So what's wrong in this figure, in a way, is that this is only true in the soma. Okay, so I inject some current. I'm injecting current in order to hold the membrane potential in the soma, but it's not true for what's happening in the dendrite, and it's certainly not true for what the synapses are feeling. Okay, this is a true thing, and especially motor neurons that are so huge. This is the neurons to do cable theory on, because they are the neurons that you'll actually get this effect the strongest. It's also true in pyramidal neurons, but that's the case. So now what they do is that they change, they change the angle of the ankle, and what you see is that there are differences when they do that. There are differences in the responses. Okay, and this is depending also on the voltage. So it depends on where so the idea here is that you change the voltage because there is some voltage dependence. Okay, this is why you do that. Otherwise, you just hold the neuron flat and will kind of move the ankle. Okay, so you want to see how is that related to the voltage, and we'll understand this figure later. Because it has to do with neuromodulation, but I think it's better to talk about what's in there before we're talking about neuromodulation. So we'll talk about the circuit, and then we'll go back to the neuromodulation if we'll have time. So last comment. Yes. Why not just holding? So there are, yeah, you can also kind of hold it at minus 60, and then at minus 50, and then minus. It will be the same thing, more or less. Yeah, it's not that important. This triangular shape is not that important in that sense. I think you're right in that sense. But it's just easy to. So the take-home message will come at the end. Okay, this is like the method part, and at the end we'll talk about what we learned from it. What you see is that there are kind of differences. Okay, so the idea is that you see differences in this response. So you see that there are these notches, and there are difference between flexion and extension. Okay, and there are also different under kind of different conditions, which has to do with neuromodulation. So we'll in a sense in a second we'll look at that. Okay. So just last comment about motor neurons. I don't remember. Did I tell you about this Indian woman that was here in? I thought I mentioned that. So when I was in a PhD, there was this woman that came to work with Idan, and she was like, her stuff was to stick needles in needles, not needles, electrodes, okay, into the nerves in humans. And mostly she couldn't actually convince many people to do that, so she was doing it on herself. But it's possible because the nerves kind of, we know exactly what they are, and the electrodes are kind of very thin, so they're not that painful apparently. You can actually stick electrode in the nerve of humans and then tell them to do stuff, right? To kind of flex or extend the fingers or the wrist or something like that, and record the activity of single action potentials in the nerve or single units. Okay, so like single axon in the nerve. And this, first of all, you can the advantage is that it's voluntary. It's hard to tell a cat to do that, but in human it's easy. Okay, and the other thing is that sometimes you can actually deduce stuff, diagnose people with that. Although this is kind of extreme because usually people use EMGs or other methods just record a much more gross activity pattern. But there are, you can go to a neurologist and he will send you to a conduction test. So people will kind of test whether the conduction of your nerves is fine. One of the things that you can see from these recordings is this size principle. The idea that you recruit stronger muscles gradually. So, for example, what you do here is that you tell the person to kind of apply some force on a force meter, all right? And then you can measure the force, and then you can also measure the firing rate of the axon, and you can see that like in low weight, you get kind of a slow response in terms of like how many spikes you get per increased weight, and then when you go to the strongest weight, when like it's in the kilograms range, then like you get a very steep curve, meaning you have like many more. So the increase in firing rate results with much more force, which is corresponding. So that on the opposite. Exactly, that's the point. That's the point, right? That's exactly the point. So you kind of so this unit is much larger. So you add like few more spikes and you get much more power. Okay, because now you recruited you remember that we talked about motor units, and they're like we kind of recruiting them with size. So now you recruit the big motor unit, and you add few more spikes, and you get a lot of power. Okay, so this is kind of that's why this scale is logarithmic. That's the idea. So for a few more spikes, you get much more power because these spikes are now going to a strong muscle, and or recruiting many fibers. Okay, a big motor unit in the sense of the same motor neuron now innervating many muscle fibers. So when you have one more spike, you get hundreds more motor units kind of twitch, motor fibers, and then you get more power.
Each line is a motor unit?
Yeah, I think it's like a single. Yeah, so these are single units. So like here, you can kind of differentiate between. You can differentiate between type of spikes. You see, this is one spike. This is one axon, and this is another axon. This is another axon. So with the same electrode, it's an extracellular electrode, and by the shape of the spikes, you can actually tell different axons. Okay, just like you do in extracellular recording in the cortex. Okay, so now we're switching to a different. So just a second. Okay, we'll start a few minutes to talk about that. Okay, so what we want to talk now about is this, first a little bit about proprioception. The things that I promised you last time, last year, right? That I promised that we'll talk about. And then we'll talk about closing the loop, okay, between the sensory and the motor. So, as I told you before, we have this dorsal root ganglion. And this dorsal root ganglion is collecting, so it's actually the place where there are the cell bodies of sensory neurons of the somatosensory system. Okay, and you remember that we talked about these stretch receptors and vibration receptors, and we talked about the fibers that are conducting these inputs and the fact that they are going up to the somatosensory system. But there's another type of fiber that we haven't talked about, and this is a sense of proprioception. So the fibers that we talked about before were fibers that conducting somatosensory input from the skin, mainly from the skin, right? You all remember what I'm talking about? Or I have to go back to the same presentation. Okay. And now we're talking about inputs that are not going from the skin, but going from the muscles. Okay? So there are mainly two type of organs that we'll talk about. One is called the muscle spindle, and the other called the Golgi tendon organ. And they actually kind of convey the information of basically of where your body is in space. What's the posture? What's the tension in each one of the muscles? And this sense is called proprioception. It's the sixth sense, if you like. It's a sense that no one talks about. That's okay. So muscle spindles are these things that are it's these kind of organs inside the muscle. So these would be the standard muscle fibers that we talked about before. Okay. And in this context of this of the muscle spindle, they would be called extrafusal spindles because they're outside, and there would be the intrafusal fibers that are inside. So inside this thing, there are also muscle fibers, okay? But they're separate from these ones, and they are innervated in a different way, and you'll see it in a second. So let's just look at that, and then we'll take a break. So if this is a spindle and you open it, what you see is that you have this fiber. This is a muscle fiber, okay, and it's looped. It's kind of wrapped by an axon that is called this 1A fiber. Okay, and this 1A fiber is very similar to the sensory fibers that are carrying the input from the stretch receptors. And basically, you can imagine that when the muscle is stretched, okay, what happens is that this thing, this fiber that is actually is just part of the muscle, it will stretch as well. Then this loop will kind of be stretched, and these stretch receptors will be open. They will carry in sodium ions, and they will depolarize the fiber, and the fiber will spike, and this spike will go back to the spinal cord. Okay, the cell body is in the dorsal root ganglion, just like together with all the other stretch receptors that are coming from the skin, and from there we'll talk about in a second what happens from there. Okay, so let's take a break and then continue with that. Yes. להעלות את השקופיות? אני אמרתי, אבל הבעיה שזה קצת... אני יכול לעשות את זה. הבעיה שאני קופץ הפעם בין שתי מצגות, ולא סידרתי את השקופיות ביחד. האם את ה... אוקיי. לא, תצטרכי פשוט לחפש את זה. אוקיי, בסדר, אין בעיה. כן. רגע, דרך אגב, אף אחד מכם חוץ מיוני לא שלח לי לינקים על המוטור סיסטם. קליפים. לא, אני רוצה לינקים, שתשלחו לי לינקים. זה היה אמור להיות להיום, אבל רק יוני שלח, אז לא עשיתי. So you can probably chop it. If not, just tell me the time, I'll chop it. Tell me between which time, I don't have a... Yeah, don't worry, I can download it and chop it.
Okay, this sounds complicated.
It's not.
I could also start at the point where I...
Okay. Send me the link. I'll deal with it. מה רצית להגיד? סליחה. כן, אבל מי sorry, so I didn't tell that, but מי שמציג לא צריך לקרוא את המאמר של הדיסקט. נו, אז מה? אז תקרא, נו, די, אל תעשה עניין. אתה רוצה שאני אספר לך כמה צפוף לי?
אני לומד 3 קורסים, וכאילו גם השני קורסים האחרים לא מתואמים, כאילו נגיד שמו 2 מטלות באותו שבוע. עכשיו אני צריך...
מה? לא, אנחנו נעשה את זה שבוע הבא, אבל תשלחו לי עד שבוע הבא.
לא הבנתי, אם אני מציג אותו יום, אני לא צריך אם אני מציג ביום אחר, כן? זה סך הכל יומיים לפני, אני צריך לקרוא אותי.
רק שנייה אחת, אני מנסה רגע רק הם ביקשו את המצגת. דבר, אני שומע.
המאמר שאני מציג ביום חמישי של
מה, טוניגאווה, נכון? זה טוניגאווה, נכון?
כן.
אוקיי.
אמרת שיש
לא היה איזה news and views? אתה... זה התפרסם איפה זה? ובאותו, הסתכלת על האישי הזה של סיינס,
כן,
לא היה כלום?
מצאתי שם את כל הסאפלמנטריז וזה...
לא, אבל לא היה news and views? בוא נסתכל על זה ביחד, עוד שנייה. אני רק לשמור את זה.
בדרך כלל זה מגיע באותה מהדורה של ה...
כל הרעיון הוא שבמהדורה, אתה הולך כאילו לפיין, אתה צריך לו current issue, כאילו לאותו issue, table of contents. ובדרך כלל, כל הרעיון הזה של news and view זה שזה כאילו מין highlight של שאתה... כאילו בגלל שזה מאמרים, אני רציתי להסביר לך על זה. זה הולך בסיין, וכל אחד זה טיפה אחר. כאילו האלה. זה האלה, זה ניוז ורדיו. סתם כאילו עושים ריוויו לדברים שהם...
שיש אחרי זה מאמרים?
אבל הם לא עושים את זה על הכל, הם בוחרים את האלה שהיו הכי טובים באותו ג'ורנל, לא לכל מאמר יש. ואז באים כאילו המאמרים. אז איפה זה המאמר?
לא היה.
אז מסגרות הבנתי. לא היה.
הנה, research article.
לא, אז פה זה יהיה, אבל זה לא היה. לא, research article זה, יש בכל עיתון, בכל ג'ורנל, כאילו בכל אישיו יש בדרך כלל שניים שהם גדולים, שהם articles, ואז כל האחרים הם קצרים וזה נקרא report. ב-nature זה נקרא articles ו-letters, report נקרא letters. זה יהיה בטח פה. אז לא היה להם את זה, יש את הפודקאסים כזה, כנראה לא היה להם. מה שעוד, אתה כן יכול להסתכל על האדיטור. אדיטור גם לא היה כאן כשאתה נכנס לזה.
לא, זה שני
משפטים, נכון, זה לא משהו. אבל מה שאתה כן יכול לעשות...
האדיטור סאמרי.
כן.
מה שכן זה מאוד, זאת אומרת, מה שאתה בהרצאה, אתה הצגת בעצם את העבודה הקודמת שלהם, שזה די אותו דבר. זה כאילו קצת הרחבה לזה, אבל זה... כן,
נכון, אז התלבטתי אם לעשות את ההוא או לעשות את זה, אבל את שניהם. אמרתי על ההוא טיפה דיברתי, אז אני גם...
המתד זה אותם מתד, זה רק... השתמשו בצבע אדום במקום בצבע צהוב. באמת, אני לא יודע למה הם החליפו. במאמר שאתה הראית, כאילו מ-2012, אני כבר לא זוכר
זה
משהו
YFP ועכשיו...
בצ'רי.
אוקיי.
אני לא יודע למה, אבל...
אתה אוהב שזה מה שנכנס להם? לא, לפעמים זה פשוט שאתה רוצה לראות עוד משהו, ואז המשהו השני הוא לראות. זה פשוט בגלל שלא נכנס לכם אווירוס. Okay, so what happened? Is the thing has a green light there, or what? No. Now it's working. It's okay. Wait. Um, okay, just before I continue with that, Oren came and asked me something which reminded me that we have never had the chance to talk about journals, and we're starting to work with the papers next week. So I'm kind of wondering if you ever been kind of exposed to the culture of journals, like what the hierarchy, which are the important journals, how does it actually work? Everybody understand that? Everybody have a good feeling of understanding what the difference between Nature and Journal of Neuroscience, biological, cybernetics? What?
Yeah, not in a very systematic way.
Nature is? תגיד בעברית.
דברים שהם כאילו מגניבים.
Okay.
זה מה שהוא אמר.
Okay.
There's
Okay.
dilution של העין and the impact factor.
Yeah. Okay. Maybe we'll devote like I'll make like 5 minutes introduction next week before we start talking about papers, because it's kind of it's something that you have to know at some point. Kind of you'll obviously pick it up from the context. What happened now? I got it on my computer. Do you get it there? This is really stupid. באמת. Sorry. Where's that? Okay. So yeah, it's something that you pick up from the air, but it's better that someone will just tell you it at once, and then it's easier to find your way out. Okay, so as we said, the muscle fiber, the muscle spindles are these organs. They have like this wrap around them, and they have muscle fibers inside of them, and they have these axons that are wrapped around them, and kind of signaling when something is stretched. What is also interesting is that on the side of these afferent axons, these IA axons, there is efferent axons, which are called the gamma efferent fibers. Okay, so the one that comes from the motor neuron, the big motor neurons, are called alpha motor neuron, alpha fibers, and they are the ones that are doing these synapses on the muscle fibers. Okay, these gamma ones, are also coming from motor neurons, but smaller motor neurons are called gamma motor neurons, and they are synapsing on these muscle fibers. Okay, what's the difference between afferent and efferent? The I and the A. Afferent are going back, and efferent are going to the muscles. Okay, so remember these. It's a bit strange to have them here, right? It's kind of we're kind of using these muscles to report the stretch. Why would we ever want to kind of also the seems to be small, not very useful. Why would we do that? And you'll see it in a second. Okay, so muscle fibers. There are kind of all kinds of details here that I'm not going to go into. Like there are these fibers and these fibers and the back fibers and the chain fibers. If you want to you can read about that in the textbook. It's not that important at the moment, but again, the gamma motor neurons are innervating the pole of the fibers just around. So this is where the afferent fiber is, and they are just on the two sides of it. We have the gamma motor neurons. Nogani, something is there. Right? Okay. Okay. So what's happening? So now you you have the muscle. You put some weight, so the muscle stretches. The axon stretches, and then if you record from the axon, from this IA axon, you'll see that the firing rate will increase. Okay. Now, if you contract the muscle, for example, by stimulating the alpha motor neurons fibers that will cause the muscle to twitch, then the muscle spindle will collapse, right? And then the tension on the stretch receptor will be relieved, and consequently the firing rate will become smaller. Okay, so this is kind of very simple mechanism to report the stretch in the muscle. Okay. So so we talked about that. We're not going to talk about that. And of course, you know, you can encode all kinds of things with that. If you stretch the muscle, like you can kind of get the response to be to kind of go together with the stretch. If you make a tap, you'll get like very quickly two spike or few spike. If you'll do like a sinusoidal stretch, you'll get like peaks around the time where you stretch the muscle, and you can release the muscle and then get a gap, and each one of these would kind of code something about the dynamic of the muscle. Of course, they are kind of like we had with sensory receptors are static and dynamic fibers. We're not going to talk about that so much here, but all this information that goes through the alpha, through the IA fibers, is going to the brain. So it's going to the spinal cord through the dorsal root ganglion, then to spinal cord, and then it goes all the way up to the somatosensory system. So it's just like what you feel from the skin, you get also input that's coming from the muscle. So your brain knows what's going on. On top of that, and differently, a little bit, not exactly completely differently, but different from the fibers that comes from the axon, this is also going to the motor neuron directly. And this is what we're going to talk about in a second. Okay, so the difference is that it goes to the somatosensory system, but it also goes to the motor system. Okay, so what will happen if we'll activate this gamma motor neuron, the one that activate here on the side of this spindle? What will happen when you activate the motor neuron? It will stretch, right? So what will happen to the stretch receptors here? They will be activated, right? There will be stretch as well. So basically, if you don't activate the gamma motor neurons and you stretch the muscle, these fibers will be stretched and will kind of increase the firing rate. But also, if you don't stretch the muscle and you just activate the motor neuron, the spindle muscles will be stretched, and this activation will go through the alpha motor neuron. When is that important?
Activate the muscle is supposed to contract or not?
Sorry. What I mean is that when you stretch the muscle, sorry, you're right. When you stretch the muscle,
it's not working when the gamma.
No, listen.
Is not firing.
Let's forget about the gamma for a second. If you stretch the muscle, you get activation of the spindle. All right. Now, assume that you don't stretch the muscle and you're not contracting it. You only activate the gamma motor neurons. Then this will stretch only these fibers in the spindle, right? There are not that many of them, so they wouldn't actually they wouldn't stretch the muscle. The muscle as itself, with all these fibers, will stay just the same. but these fibers inside the muscle will be stretched.
That's what I'm asking. Why do they stretch and not contract?
So because what happened is that, okay, so you're right. It looks like this. You're right. It looks like it should contract, but actually what?
I know about
Yeah, no, you're right. You're right. But it's confusing. You're right. So these fibers are kind of bundled together with with all the muscle. Okay. So now if you kind of contract this side and you contract this side, then the middle part will be actually stretched. Sorry, I didn't explain. Right? You following? So basically, you yeah, okay. So we have a muscle, and inside the muscle we have the spindle, and inside the spindle there are these fibers, and they're wrapped around with this alpha fiber, which has stretch receptors. Now if I put weight on the fiber, the muscle is stretched. These muscle fibers are stretched as well. The whole thing is stretched. And then the alpha fiber is stretched, and then there is an increase in the firing rate of the alpha fiber. Now assume that the muscle is at rest. It's just sitting there, nothing happens. And now instead of stretching the muscle, we are activating the gamma motor neurons. So the gamma motor neurons are actually on the two sides of the fiber. So if they are activating, this side will be stretched, and that side would be contracted. This side would be contracted. The other side will be contracted, and they are actually held by the same, by the muscle. So the middle part will be stretched, and then this will code as well for stretching.
So how is it activated by contracting the muscle?
You're not contracting the muscle. When you contracted the muscle, it's not activating. It's the opposite. It's not activating. It's reducing activity. When you activate the muscle itself, not the gamma motor neurons. When you are activating the alpha motor neurons. The alpha motor neurons are contracting. The alpha motor neurons are contracting the muscle, and then the stretch here is relieved. Okay, and then so it sounds strange, right? And so here's a solution to this strangeness. Okay, let me explain that, and then everything will become clear. Just 1 second. Okay, look. So if you stretch the muscle, you stretch the spindle, you get elevation in firing rate. Okay? No, now you stretch the muscle. You put a weight on that. You stretch the spindle. You stretch the alpha fiber. You get elevation in firing rate. Okay. Now, if the muscle is like relaxed, then the spindle is relaxed. Okay, and basically you don't get any activation from the spindle, right? Because it's now there's no stretch on the stretch receptors, right? You see it here, right? It's kind of midaldel. Okay. So now suppose you now want to be sensitive, even though the muscle is kind of in a resting position, or even kind of contracted, and you want to get some input from the spindle. So what you can do, you can activate the gamma motor neuron. The spindle will be stretched without affecting the muscle. So the muscle is still in the same position, but now the spindle is stretched. So now any small stretch to the muscle, you'll get activation from the spindle. Right? So basically, it's if you like, it's a gain control mechanism. So you shift. So this is a response of the spindle to stretch. Okay. But now when the muscle is kind of contracted, you are out of you're kind of in this regime, because the muscle is now midaldel, because it doesn't feel anything. So you stretch it back, and you kind of move the response curve to the left, and now it's kind of you again somewhere here, so any difference in the stretch of the muscle will be encoded. Clear?
Does it happen naturally?
Yeah, of course. What do you mean it doesn't? No, it doesn't happen naturally, or no, you don't follow? It's not clear. Okay, I'll explain it again. It happened naturally in the sense that
it doesn't move.
Yes.
Why?
Because you want, because of that. Exactly
No,
because
why?
of,
Why does it happen naturally? In which scenario?
in the scenarios that your body is like when you're sitting down and after half an hour your hands are like that. Your body wants to know that when you kind of move the muscle, but when you do that, your muscle is contracted, so your spindle wouldn't do anything. So unless you stretch the spindle to be kind of responding, when you move the muscle, you wouldn't feel it. So it's like it's a kind of a feedback control mechanism that the body is using in order to be sensitive all the time to what the position of the look. This is like a general mechanism. It's an example here, but let's take another example. Okay, there's examples that you should know because I think Shaul told you that, and I certainly cannot do it right in terms of I wouldn't be accurate now because it's from memory and I haven't prepared that, but just as a kind of from like it's almost trivial. Suppose you have a photoreceptor, okay? So what does a photoreceptor does? Right, you have a current and you have light, right? And with no light, you don't get any current. Okay, actually, photoreceptors going the other way around, right? But just for the sake of like thinking about it, with no light, you don't get any current, and light increases, you get more current, and then they saturate. If you kind of pull a lot of light on the photoreceptor, it will saturate. Okay, it cannot produce more current than the maximum current that it can, right? But so this is what we see here. Now, if I'll switch the light off, okay, at the beginning you don't see anything, right? Because it's dark, and then you kind of adapt to the darkness. So, we're talking about, say now, only cones. We forget about the fact that we switch to rods and stuff. Okay, I'm not closing completely the light. I just switch it by, from 10 lamps to 1 lamp. There's still light. You can still see, but at the beginning you'll have darkness, right? What happens is that now this is on the scale of these 1 to 10 lamps. So this is 1 lamp and this is 10 lamps, and you kind of this saturate and this is 0. Okay, but now we are actually working in this regime. Okay, so we can't see anything. So what actually happens? There's this kind of the adaptation to light, right? This curve will actually move here and will do that, right? And now with this 1 lamp, you're here, and if you increase the light a little bit or decrease it a little bit, you still kind of can encode it with the current. Okay, so this is kind of the adaptation. You're still not with me. I feel that you're not.
So the point is that if I activate the gamma motor neuron, I will be more sensitive to small changes in the muscle
Exactly.
and in the muscle, and that's totally related to proprioception.
Yeah. So this mechanism is a very general mechanism of adaptation. You want to be on the curve of your response curve that small changes will give you big responses, or at least will give you some responses. If you are sitting here, okay, any change in stretch will not give you any response. So this is not a good place to be because you can just chuck away this muscle spindle. It doesn't do anything. Okay? So what the gamma motor neurons are doing, they're kind of moving, they're stretching it artificially. Okay? And they're moving, which kind of moves the response curve towards here. And now when you're here, again, changes in the stretch of the muscle will be encoded in spikes. Okay? And you do that by artificially changing the muscle spindle. Now, the context is exactly what I said before. Okay, so if I'm standing like that, okay, so my muscle is stretched to some degree. Okay, and now if I'm moving the hand the muscle, this muscle is stretched, right? And this will be conveyed through the alpha fibers, the 1A fibers, sorry, to the spinal cord and to the brain. But if I'm sitting next to the computer and I'm like that, then my muscle is stretched to a different degree, okay? And now if I will kind of use the muscle spindles coding for this regime, I would not get any change in activation when I do that, right? So I kind of adapt. So my brain now knows that I'm sitting like that. So what it cares about is the movement around this point, and not around that point because I'm not there. Okay. So I'm changing the dynamic range of these muscle spindles to encode relative to the position that I'm at. It's like as if you have like two. You can think about it as if you have like two memory or two. So one thing encodes, the general position, and then the other one encodes what happens around this position. Okay, and at the beginning, if you track all the time what you're doing, you can actually keep track on these two things separately. Okay, but if you have one muscle spindle that is always the same, then you lose your dynamic range. You can encode only around one direction and be much less sensitive. I mean, what you could do, obviously, is generate a muscle spindle that has like a way bigger dynamic range. Okay, so you can make like, I don't know, hundreds more wraps of this IA fiber, but then you lose the sensitivity. So you can't have them all. So usually the trick in many sensory systems is to, we'll have to jump now over many things. But how does it what happen?
More activation of the gamma.
So this comes from central system. So this is kind of what you see here. Okay, so basically the motor cortex, if you like, or some other parts, like subcortical areas, but anyway, higher areas, are sending a command to the muscle. Okay, and then you get responses through the spindle to the motor neurons and also to the cortex. But at the same time, there is commands to the gamma motor neurons, because the brain knows that it's going to contract this muscle. So he knows that this is going to stretch, and it knows that if you still want to be sensitive, you need to activate the gamma motor neurons in order to compensate for that. Okay, or sorry, to that muscle.
You can't do it with intention.
To activate the maybe if you are like an Indian monk, you can. I don't know how to activate my gamma motor neurons, but maybe we can do like biofeedback on that. It's a good question. Not intentionally that I know. I mean, I think it's a kind of an automatic system that happens there. It's like you can say that you don't know how to activate your alpha motor neurons, right? But you do, in the sense that you know how to move. But it comes together with that. It's not like specifically. Yeah.
But if we stimulate the alpha motor neuron, then you talked about it before, but it doesn't clear, the muscle will
Contract.
stretch or contract.
So look at that. Okay, so you activate the alpha motor neuron, the muscle contracts. Okay, and then in order to be sensitive, now after it contracts, because the load on the muscle spindle now is reduced, you have to activate the gamma motor neurons in order to keep the spindle stretched. So you do that in parallel. Okay. Okay. Do we have time? Okay, so I'll just mention that there is another organ which is called the Golgi tendon organ. And you know that if these are the muscle fibers that are kind of bundled up together and held by the tendon, הגידים בשני הצדדים של השריר, and then inside these tendons is another type of fiber. It's called an IB fiber, which is kind of like making a mesh inside the tendon. And then again, when the when the tendon is stretched, it's sending input to the spinal cord, and I'm not going to. So the idea is the same, so it's really not kind of that interesting to go into the details. No, really, I kind of we won't have. We need to just at least mention reflexes in the 2 seconds that we have for that. So, question.
So how does the IA fiber know where the gamma neuron is actually corresponding?
What do you mean? Someone told them. What? I don't understand.
Hardwired in development?
It's hardwired completely. Yes. It's hardwired, and it's going through. That's the kind of the thing that we're going to see in a second. That this fiber that goes IA, it goes to the same motor neurons that are activating. It's the same muscle. So it's hardwired. How does that happen? That's a miracle. That's completely a miracle. I don't know. If I had to design that, it would probably won't work. Okay, so spinal cord is divided into segments. We talked about it, and segments are corresponding to these dorsal root ganglions, and there is this mapping that lower segments are innervating lower parts of the body, and higher segments are innervating higher parts of the body. And if you cut through the spinal cord, what you see is this gray matter, and this butterfly shape is changing through the segments. I think we talked about it last time, and it's partially because it's kind of somewhere here in the middle of the body. You have to have like a lot of fibers. That some of them are going all the way down, some of them are going all the way up. And toward the end, you have more. You don't have so many fibers that are going up and down, but you have like motor neurons that has to control the limbs, and they are bigger, and you need to have more of them. So if you cut through you'll see that the butterfly is kind of changing the shape, but the general structure is very similar. And you have this dorsal part or posterior horn, if you like, and the ventral horn. And in the ventral horn, you have the motor neurons. And what we're going to talk about right now is actually how these loops are closing. Okay, so we talked about it many times. You have like this alpha IA fiber coming through the dorsal root ganglion. They're going through the dorsal root, and then they're doing 2 things. One, they're going up to the central nervous system, and second, and also a little bit up and down in the spinal cord, and second, they're crossing to the dorsal root, and they're innervating neurons in this to the ventral root, and they're innervating neurons in this ventral root. So you'll see it in a second. If you look along the spinal cord, you see that they are going in, and they're kind of going up and down in the segment, and they're kind of innervating along the spinal cord. And the key thing that we had to at least mention here is this reflexes, and this was what was funny. So this has been found by Sherrington. I think he actually got the Nobel Prize for that, but it's very long time ago. And this is a stretch reflex that I guess everybody knows, but the idea is that you tap on the tendon here, it stretches the muscle, so the muscle is stretched. Okay, and then when this muscle is stretched, we know that the muscle spindle are stretched. The information going through this IA fiber that the cell body are in the dorsal root ganglion going into the spinal cord through the dorsal root and making a synapse onto motor neurons that are innervating the same muscle. And when these motor neurons are activated, the muscle is contracted. Right? So you stretch the muscle and it's contracted back. And this is the thing that your doctor will check when they're doing this tapping on underneath the knee. And this is a monosynaptic loop. Okay, it's like there's one synapse here, and this is why I told you that this results with this intracellular recording doesn't make a lot of sense, because this is a case where you don't stretch so many alpha neurons, IA neurons, right? You stretch the muscle a little bit. It's actually a very small thing, right? And you get activation of the motor neuron that contract the muscle. So it doesn't make much sense that you put an electrode on the eye muscle and you kind of stimulate them very hard, and you get this subthreshold activation that doesn't produce any spike. The reflex wouldn't work if that was the case, right? You're with me? Okay. So, but this is the simplest reflex you can think of. Sherrington went from that reflex to think that basically the whole brain is one big reflex. That everything is like that, and you think it's funny, but it's actually exactly what it is. It's turtles all the way down. If anyone knows what I'm talking about, but this is like the simplest neural networks that you can think of. It's one neuron connecting to another neuron that is kind of closing the loop and that thing. And now you can basically what's coming on top of that is more and more complicated reflexes. We'll see them in a second, or at least one or two of them, and that's it. That's everything you need to know about the brain. It's in a way surprising in that it works like that at all. You know, this is kind of when you think about how the brain works, you think about very complicated things, right? And this is a control loop which seems to be very simple. There's nothing to understand here, and but it's there. It is being used, and it's there. Now, whether this is the whole thing, it's a bit kind of far-fetched, and we'll talk about that in a second. Whether it's so simple, whether this is, for example, whether this is linear or not. Okay, so one of the things that people have shown, use this system to show is like whether two synapses are summed linearly or not. Okay, for example, because you can take split this fiber and kind of stimulate one type of IA fibers and another type of IA fibers. You get two EPSPs. You get from each one of them, you get an EPSP, and then you activate them together and see if you actually get the linear sum of the two. Okay, and the result was that you can actually get the sublinear sum. They don't summate linearly, and you know that because of the reversal potential and stuff like that. But it's less relevant here. But this could be. This will take us in a second to where we started. So stretch reflex. Okay, there's one more thing to think about. Is like this is a reflex, but we'll go back to that in a second. Okay, so this is one reflex, and then there is another reflex, and in this case, what happens if you activate the IA fiber of the other neuron? So if you activate, so this is what you stimulate the fibers from the muscle, okay, and you have the one synapse of the motor neuron, and it goes to contract the muscle, okay. But what happens if you activate the IA fibers that coming from the antagonist muscle? Okay, so this is like if I stretch this tendon, this will contract that muscle. What happens from that muscle? Okay. So these muscles also go, and synapses now not directly onto this motor neuron, because then you'll get exactly the opposite thing, right? The synapses on interneuron that is inhibitory. Okay, so the antagonist muscle is actually making inhibition on the motor neuron of its antagonist muscle. Okay, so now you see that it becomes a little bit more complicated. Okay, and you have and if you record the responses, so if you stimulate that one, you get right after you stimulate it, like within less than a millisecond, you get an EPSP. And if you stimulate that one, it will take a little bit longer because it goes through another synapse. You have to activate this neuron. It goes through that synapse, and then you'll get an IPSP, and it will have a delay. So this is what's called the disynaptic reflex, and this is a monosynaptic reflex. But it could be like, you can have like 100 synapses. So one of the things that is obviously kind of wrong in this picture is that it's usually not enough with one synapse to activate one interneuron that will then activate or inhibit the interneuron, right? It's like you need few fibers here, at least, in order to excite this interneuron, and then it becomes a little bit more complicated because not all of these fibers are coming from exactly the same spindle and stuff.
But in the previously, one synapse was enough.
Here, so again, it's not one synapse. I think if you take one IA fiber from a spindle, just one, and activate it, you wouldn't get the reflex. You need more, and you have more. You have a lot of spindles in the muscle. Okay. So we can jump that. Okay, so there's one more reflex that might be interesting, and mainly because of this cross. Okay, so you see that the inputs are coming, and these are A delta now. They're not fibers from the muscle spindle. There are fibers that are coming. I told you that not only the IA fibers are synapses on the motor neurons. In this case, it's I delta, which are actually coming from nociception. So like pain or C fibers. I think these ones are not C fibers, but and the idea here is actually to create a reflex where you, for example, touch a hot plate, okay, and then it will just move your hand, and then what they do is they stretch the extensor muscle and inhibit the antagonist muscle. Okay? So you get the movement that you want, and
it goes to the other side as well.
Because you need the two.
No, it's the other leg.
Yeah. Yeah, exactly. I think so. It's like, well, I haven't thought about that. You're right. Yeah. I need to think about this. I actually didn't think about it. You're right. Yeah, so they're activating both the extensor and the flexor, and they're also controlling the other side of the body, probably to do something with your balance. And this is also coming here. So the main point, and that's the point that we're going to talk about in the next lessons more about, at least try to, is that all these reflexes seems to be completely automatic, right? You you stretch the spindle and it kind of stretches it contracts your muscle. So this kind of doesn't leave a lot of. So just imagine that you try to walk, okay? So you walk, and your brain is making a command for your leg to move. Some muscles are stretched, right? Okay, so this muscle is stretched, right? When I'm doing this movement, and then immediately this muscle is stretched, and then immediately the reflex will kind of contract it. And my leg will do the opposite thing of what I want to do, right? So you can't actually move if everything would just work on reflexes like that. So they're kind of doing all these things. So you have to have a central control on that, and this is kind of the essence of the motor system, that you have like local control, and then on that you have higher order control and higher order control, and it becomes very, very complicated. And the things to kind of consider here is that at least in the stretch reflex, there is not much place where you can actually do this control, right? Because this is coming immediately from there. You cannot really control that very much. It comes from the tendon. The only way you control that is what? Is a gamma motor neurons, right? You can actually play with the sensitivity of the tendon, of the spindle, right? You can then somehow try to, so there is no inhibition on that, but you might actually have some inhibition that come and kind of will do something here. We call it presynaptic inhibition. Inhibition directly on the axon that are going to the nerve. Okay, and that's the point where you can actually interact, or you can have some inhibition going directly on the motor neuron. Okay, on the motor neuron, on the axon of the motor neuron. But these are the places where you can interact. Okay, there's one motor neuron one neuron that I kind of why is it here? I kind of skipped. Why is it here? This is this rational neuron that we kind of ignored. But you see, there are these neurons that the alpha motor neuron are actually excite, and there are inhibitory neurons, and they are kind of inhibit other neurons. So you have all kinds of feedback things that can help you, can help you get rid of the reflex when you don't want it, in a way. Okay. So I mean so that what?
These are controlled from the central?
So I think the idea is the way I understand it is like if you don't do anything, right, then the reflex is intact. But when you plan to do a movement, you get rid of the reflex so it doesn't get in your way when you want to do it. So it's a complicated mechanism in the sense that you can activate a circuit that will even do something on its own just due to reflexes. So there are systems like if you take, I'll give you an example. When you look at a fly, okay, a fly has or a locust. I think in locust it's even better. So locust are chagavim, okay, and they fly and they do this kind of. They have to move the wings. The muscles that are moving the wings are very strong and they're very big because they have to create a lot of force. And basically, they have like a system that are kind of a gap junction between them. So one contract, it causes the other to, they're kind of having an oscillation between them with no neurons. Okay, the one is contracting, the other one is relaxed, and the opposite. And they're working against each other, and this is what makes the wings move. And then you have like small muscles that kind of control the angle of the wings in order to fly. So in a sense here, you also kind of, the idea is that you want to engage like central pattern generators that will kind of do the walking for you without you thinking about it. You don't want when you walk the motor cortex to be thinking of every step. Otherwise you wouldn't get anywhere. Okay, and but then you want to have the control, like first of all to send this command, and then be able to kind of tweak it when you want. So it's kind of this kind of hierarchy that you have there. Okay, so just as an example for that, this is an example of you're making a task and you concentrated on this muscle here. Okay, and what you do is that the person is standing in this position, and it holds a handle, and the handle kind of pull the person towards the handle, so in this direction. Okay, in these two cases. But in one case with this hand, she holds the table, and in the other case she holds the cup. Okay, and when you perturb with this pull, and you have a table, what you'll do is you'll push the table in order to hold yourself in place. But when you hold the cup, you wouldn't push the cup. You actually will pull the cup in order to what? In what sense? No, this is not voluntary because you don't know when it's going to happen. You stand, you hold the cup, and suddenly you do that, and this is a reflex. You don't think about it. You don't have time in order to you might think about it, but you wouldn't have time to react properly. You'll fall if you think. Okay, so this is happening in a reflex. But what's nice here, it's your brain actually knows, in what position. So your brain knows that if you push the table, it will help you. Okay, and it knows that if you push the cup, it wouldn't help you. So the idea is that there is here, and this is what happens, right? So it's like the activity in the muscle is different. Okay, in one case the muscle is doing that, in other case the muscle is doing that. Okay, so the idea is that you have a reflex, but the direction of the reflex is controlled from higher order brain areas. I don't think that we know exactly how that works, but that's the idea. Okay, and this is brings us back. Yes.
Is it only the reflex in the arm? Because when I think of this scenario, it's more like leaning your whole body against the table because it will support you, and then the muscle is doing what it does, or just leaning your whole body to the back because otherwise it will just fall over.
But whatever, I see what you mean, but what I'm trying to say, it's not me who's trying to say that, but it's a whole, yeah. So it's true. It's a whole part. It's a part of a whole kind of posture thing. So it's like the brain is not okay. So the brain in that situation is not just controlling the reflex for this muscle. It's actually sending commands to many neurons and many places to control all the reflexes. And of course, if you'll do a different perturbation, your body will be prepared for many of them. Obviously, there are perturbations here that no reflex will help you. You can create situations that you will do the wrong thing. But in many cases, to our surprise, is the reflexes are doing something different than what we would expect, because somehow your brain knows how to deal with this situation, and some of that thing require learning. Okay, so for example, the classical cases are like when you learn to skate or to ride a bicycle, and your first reflexes are doing the wrong thing, right? You feel you want to fall, and you start doing that, and you actually fall more, right? And what you learn to do is actually to control that and not do what you actually want to do. And once you learn to do that, it doesn't happen. It happens all the way. This learning it happens in the reflexes, happens in the brain, and eventually it becomes kind of a reflex. That's why I said, you laugh, but it's true, because when you eventually skate, you don't think of that at all. It's becoming a complete reflex. Or when you do, it's the same thing when you do martial arts, right? So you learn how to avoid things, right? You learn how to avoid movements that wouldn't help you. So just yes.
And your definition of a reflex is something you don't think about? Because I studied
Well,
that involves
it's a good question. What?
I studied just closed circuit goes through the spinal cord and doesn't involve the brain. So
Yeah, I think like to be an accurate definition would be something like a circuit that you can a relatively simple loop type of circuit that that is generating an automatic movement, something like that. But that's what I said when I talked about the fact that Sherrington was trying to imply that the whole brain is one big reflex. Is that where do you stop? Like if you have dysynaptic reflex, it stops, or if you have like 3 or 4, how many? When do you say it's not a reflex anymore? Okay, and this is not a good answer to that, I think. Okay, so just last bit. So the point that was with this slide. Do we have time or we? No. Okay. Okay. So I'll just mention that in the next, like first time in the next time. Sorry. תקרא אחד ולא שניים. זה מאמרים מאוד קצרים. אחד תקרא, אחרת אתה לא תבין על מה אנחנו מדברים, כי זה לא קשור לשום דבר שדיברתי בכיתה.
גם המאמר שלי, כאילו לא רואה את זה קשור לגליה בדיוק.
מה זאת אומרת איך זה קשור לגליה? זה אסטרוצייט. זה אסטרוצייט, זה הנוירונים שעושים את המייליניישן. זה גליה. מה? הם מדדו מהאקסונים, אבל מה שהם מראים זה שהם עושים את זה הם עושים מניפולציה באטרוצייט. מה זאת אומרת, אתה לא מבין איך זה קשור לגליה? אבל תסתכל מה עוטף את האקסון של הפירמידל סלס ואיפה הם עושים את המניפולציה שלהם. כדאי אם אתה מתקרב לפרזנטציה שלך ואתה עדיין לא מבין
שזה... לא, קראתי, כאילו יש שם כמה עמודים, קראתי את העמודים הראשונים ולא הגעתי לזה.
אוקיי, הלו. היי. Again? Yeah, I'm coming. I was teaching. I'm coming down in 2 seconds. Okay, I'll be in the lab in 3 minutes. Bye. אני לא מלמד השבוע. עד הפעם הבאה. הפעם הבאה זה שאנחנו מדברים על הסומטו-סנסורי. יש איזה סשן אחד, נכון? זה הולך עם הרמפה. מה?
על הומו אני מדבר.
אתה מדבר ואז אני אדבר, נכון? אתה נותן לי איזה שעתיים או שעה. אוקיי, ואחר כך בסוף יש לי כמה שעות.